Hello everyone. Today we will be talking on Staphylococcus aureus, its virulence factors. I have started a series on Staphylococcus aureus and I am discussing its importance with different headings. Today I will be talking about its virulence factors. So, what is a virulent factor or what is virulence factor? It's a pathogen's ability to infect or damage host tissues and cause discomfort to it. So, a pathogen enters inside the host and it can cause discomfort to that host by manipulating its innate or adaptive immune response. What is innate and adaptive immune response? That I have taught in my previous lectures. If you want to know more about it, you can go to those videos. So here we are talking on virulence factor, that too of Staphylococcus. So these are the factors which are responsible for causing or making Staphylococcus aureus an infectious organism or an organism which is a problem for us. So what are the things it's carrying that is responsible for causing discomfort to us and creating problems? So that we will be discussing in the next few slides. And these virulence factors also help the pathogen to survive in hostile environments. So, what are these different virulence factors of Staphylococcus aureus? One is cell-associated polymers. We will be talking about them in detail. Other is cell surface proteins, exoenzymes, and exotoxins. So, coming to first one, that is cell-associated polymers. That is the mostly in the cell wall or the total structure of cell wall which is present in Staphylococcus. So we will talk about three layers of Staphylococcus aureus that is responsible for it being so organism that causes so much of discomfort to us. So in first the three layers, first is the cell wall, another is teichoic acid, and third is capsular polysaccharide. So this cell wall will be surrounded by capsular polysaccharide and will have teichoic acid embedded in it. So we coming to cell wall, how it is causing problem to host is that we all know that cell wall of gram-positive bacteria, and that to Staphylococcus aureus, which we are studying today, consists of polysaccharide and thick peptidoglycan layer. So this layer protects the organism from getting in touch with complement system. If complement system gets activated, it may be opsonized, it may be kills. What is opsonization, what is complement system? If you want to know these things, you have to go and see my previous videos which where I have talked in detail about this thing. So here I'm just naming the things that complement system gets activated and protects human or an organism from the attack of pathogen. So this pathogen is having a thick cell wall which is protecting it from getting recognized by complement system, and this cell wall is also protecting the bacteria from getting by getting killed by or by getting lysed by osmotic pressures. So this cell wall is protecting it and getting it a entry pass into the host and disturbing the host. Another thing which is embedded in this the wall of this bacteria, cell wall of this bacteria, is teichoic acid, which is highly charged. So this highly charged teichoic acid helps in adhesion of cocci to the host surface. Or if an implant, if we are implanting any prosthetics inside the body of a human, then this can form a biofilm and can disturb the host. And this teichoic acid also protects the bacteria or cocci from complement mediated opsonization, same thing that cell wall was doing. Now, coming to the third part, that is capsular polysaccharide, which is present in this bacteria. This also masks this bacteria from recognition by phagocytic cells, because patrolling cells are moving in our body, which can phagocytose the foreign particle or foreign thing which is traveling inside the bloodstream, but or tissues. But this Staphylococcus aureus is able to fool or mask the phagocytic cells by this capsular polysaccharide. This is a protective mask this bacteria is wearing. Another virulent factor is cell surface proteins. These cell surface proteins are present on this bacteria, and this is named as protein A. It is also called as B cell superantigen. Why B cell superantigen? So B cell superantigen means it helps in proliferation of B cells, high multiplication of B cells, which is not required. It is anything which is more than required is also a problem. So this protein A is chemotactic in nature. It is anti-phagocytic. That means it can fool phagocytic cells. It has anti-complementary effects. Complement system is another protective system by host, but it is fooling that system also. It damages the platelets also, induces hypersensitivity, and participate in coagglutination. So if coagglutination term, hypersensitivity, you want to get clear, you have to go to my previous videos. Then next virulent factor is exoenzymes. Enzymes we all know we know what are enzymes. Now, what are the exoenzymes this bacteria is having, which helps this bacteria to survive in the host cells and cause discomfort to host, while this bacteria can easily reproduce and take nutrition from the host cell. So these exoenzymes which this bacteria is possessing are cofactors, lipases, hydroneuridase, nucleases, which was originally called micrococcal DNAs, and it is also called thermonuclease because it can survive, it can resist high temperatures, and it also has proteases. So these are the 5 exoenzymes this bacteria is having, and what these exoenzymes are doing, let's see one by one. So what are these cofactors? Cofactors are the things which utilize the zymogens of the host. So this needs host zymogens for activation. So it will go inside the host and use the host machinery, that is zymogens of the host, to get activated. So what are those cofactors present in the Staphylococcus aureus, which will be using host machinery to get activated? Those enzymes which are present in Staphylococcus aureus, or the cofactors which are present, we are calling them cofactors because they will need zymogens of the host. They will need host machinery to get activated. So what are those which are present in Staphylococcus aureus? One is coagulase, another is von Willebrand factor binding protein, and the third is staphylokinase. So you can see the short forms of these. These are the 3 cofactors here. These are basically enzymes, if you see the name. But why we are calling them cofactors here? Because they will be needing zymogens of host to get activated. And once they are activated, they will manipulate host innate defenses. What is innate defenses? What are innate defenses? Again, you can go to my immunology lectures and get it clear. Once it is clear, then you will know that how these cofactors manipulate the innate defenses to promote bacterial survival and dissemination. Their number will increase because this way they will be taking the host machinery, using it, getting nutrition from it, and then they will multiply in number and disseminate, become more infectious. Another exoenzymes are lipases, that I have already named you, and as the name suggests, they will be attacking those they will be using the zymogens of the host in particular cases, and all these enzymes will damage host tissue, derive nutrient from the host tissue, and will multiply and disseminate. Another type of Now, we were talking about exoenzymes. Another type of virulent factor is exotoxins. So we have already uh, talked about if we will consider, if you consider the structure of Staphylococcus aureus, you will see that it has a wall which will protect its protoplasm. So wall have three structures, three layers which are helping it to cause infection or become virulent in the host. And these three structures we have described one by one in detail. That was cell wall and polysaccharide capsule, and the third one was teichoic acid which is embedded inside the cell wall. So these three we have already talked. Now coming inside the bacteria, we have talked about the outer wall of the bacteria. Coming inside the bacteria, we have talked about exoenzyme, the enzyme system that bacteria will be having. And now we are talking about exotoxins that bacteria is having. Exotoxins are again the toxins which will be secreted by bacteria using host machinery. So what are those toxins? One is cytolytic toxin, another is superantigens, and cytotoxin enzymes. Now these cytotoxin enzymes are the enzymes which will be damaging mammalian cells, and a kind of cytotoxin will be secreted in this. So cytotoxin is, we will not be going into much details of cytotoxin enzymes. We will talk about cytolytic toxins and superantigens. These cytolytic toxins are hemolysins and leukocidins. You can know by the name that what are hemolysins. They will be lysing the heme part of the host, and leukocidins will be damaging the leukocytes of the host, because cidins are killers. So cidin mean killing, and cidin is killing. So leukocidin is killing leukocytes. Same with lysins, will lyse the lysis will be again destroying the heme part. So hemolysins and leukocidins, they are four in number: alpha hemolysin, beta hemolysin, gamma hemolysin, delta hemolysin, and leukocidins will be there. And what all these cytolytic toxins will be doing? They will be destroying host cell membranes. Now what they are going, till now we were talking about the way in which the bacteria can enter, and it's like a terrorist which is hiding in different, masking itself. And now the through the mask and through all this teichoic acid, by that means it was charged through and it adhered to the cell of the host. So now the terrorist, that is the bacteria, or the invader, that is a bacteria, have used all the mechanisms to cover itself and enter inside the cell. And once it has entered inside the cell, it has started using host machinery for its growth and reproduction, taking the nutrients, and that was done through exoenzymes. Now these toxins are doing what? Now they have started damaging the cell membranes of the host cell, thus resulting in lysis of the target cell and inflammation, causing inflammation to the host cell. What superantigens are doing? They are again the type of superantigen here we are discussing. And if you want to know more detailed things about superantigens, not only of staphylococcus, but what generally superantigens are, you can go to my video. I have posted it, and there I have talked about superantigens. Here I am focusing on superantigens of Staphylococcus aureus. So here the T cell superantigens, which are produced by S. aureus, and it's the largest family of exotoxins of S. aureus. They are extremely stable, highly toxic in humans. Different kinds, when we will be talking about pathogenicity of Staphylococcus aureus, and that I will be discussing in my next coming lecture. In pathogenicity, we will go into the details how this T cell superantigens are damaging the host. Then now here we are focusing on types of superantigens which Staphylococcus aureus possess. So one is T cell superantigens, second is enterotoxins, third is enterotoxins like superantigens, and fourth is toxic shock syndrome toxin 1. So these you can, if it is asked how many types of superantigens are present in Staphylococcus aureus. So you can name all these four. Then the role of these superantigens is to mediate massive cytotoxic production, trigger T cell and B cell proliferation. And when we will be talking about the pathogenicity of Staphylococcus aureus, there we will discuss all about the details how these superantigens like toxic shock syndrome toxin is helping bacteria in invading the uh, host cell. Here I have just listed the virulence factor. Hope you are going to like this. Thank you so much.